Thank you, everyone, for staying through the morning. My name is Matthew Savary, as Dina said, and I lead the MedTech side of the New Ventures Group. We have something called the Innovation Fund, and we have got projects from the Innovation Fund that we want to show to you today. Before I move into that, just as a means of introduction, I'm sure that you're wondering, what is the Innovation Fund? So the way that I imagine it is the Innovation Fund is a tangible manifestation of UCLA's commitment to innovation and bringing really promising technologies to the patients where it matters. That doesn't sound like a ton, but I'm going to tell you something that everybody already knows, in that the years before the next big thing becomes the next big thing, it's a really great idea that's sitting in a lab, written up in a really esoteric journal, and it takes a lot of money, a lot of time, and a lot of resources to take these things all the way to market. And before we can get these things attractive enough to you good people out here, really frequently they need to establish some levels of milestones and progress so that you can be confident that it's something that's worth investing in. And so the best way to tell you about how that actually works is the UCLA Innovation Fund invests $200,000 or up to $200,000 in what we'd consider really promising technology, non-dilutive funding, just as you're aware of that, so that we can move these projects forward and start hitting milestones. And so without further ado, I give you Drs. Moshe Varinia, Dr. Hubschman, and Dr. Bernthal showing you the next big thing.
Thanks. So thank you for the invitation and the opportunity to share with you our project on cataract robotic surgery. So cataract surgery is the most frequently performed surgery in the US, with more than 3 million surgery every year. And despite some great progress in laser technology, we still rely today for traditional cataract surgery on optical microscope with a limited visualization of the capsule of the lens. And we think that using a robotic system with real-time image guidance will help us achieve better surgical outcomes. So the team leading this project is myself. I'm eye surgeon here at UCLA at the Stein Eye, and Su Shin Tsao and Jacob Rosen are both professor of engineering here as well at UCLA. So cataract is very frequent. More than 50% of the American people over the age of 75 will develop a cataract. Cataract being, is the cloudiness of the lens, which is here, and will impair the vision. It's a huge market. Cataract surgery represents more than $4.5 billion every year, 8 million surgery worldwide, and this market is supposed to keep growing pretty quickly. Cataract surgery can be split or breaking down in 3 different steps. One is the access to the lens, the second is the lens extraction, and the last one is intraocular lens implantation. Today, with traditional surgery, we can use laser technology, femtosecond laser, to achieve in an automated fashion the access to the lens. However, the lens extraction today is still manual, and the surgeon is manipulating the tools inside the eye with a high risk of breaking the transparent capsule of the lens. So what we've done here at UCLA with my colleagues, we have developed a robotic technology using real-time OCT guidance, OCT being optical coherence tomography, which can really visualize all the part of the eye very precisely and use this information to guide the system and remove all the pieces safely. So cataract surgery, again, 3 million people every year, but there are still some complications, as I mentioned before, which is mostly the posterior capsule rupture, which happen in 1 to 5% of the surgeries. And this complication leads to other complications such as retinal detachment, macular edema, and everything. Now, when you try to be very careful and remove the lens without touching the capsule, you can leave some material inside the capsular bag, and this leads in more than 50% of the patient within 5 years after the surgery to posterior capsular opacification here, which lead to limitation of the vision, and which will require secondary treatment with laser. So we really think that there's an opportunity to improve the surgical outcomes using robotic technology and real-time image guidance. So our technology includes different parts, but So we've designed the robotic platform. We have many features, but as I mentioned before, the main one, I think, is the automated capability using real-time guidance to remove those pieces safely. So how does it work? We have a real-time OCT acquisition. So this is the inner part of the eye, the cornea, the iris, and the lens. So when patient develop cataract, we need to remove this by using aspiration and so on, but making sure that the capsule here is safely maintained. So here during the surgery, we're using our technology. The OCT will scan the inner part of the eye, as you can see here, the iris, the capsule, and the lens, and over time, will remove all those pieces safely. So we have developed technology allowing us to segment the information in real time. So acquiring the OCT is not that difficult, but segmenting in real time and recognizing all the different structure of the eye in real time is tricky, such as the cornea, the iris, the lens, and so on. And so we can develop a predefined trajectory within the lens, as seen here, and this trajectory of the tooltip will be adjusted on the fly depending on the modification of the anatomical structure during the surgery. So we've done some initial evaluation on harvested pig eyes. The surgical time was pretty much the same as manual or traditional surgery. No posterior capsule rupture was seen during this evaluation, and 100% of the lens was removed in 25 over 30 eyes we tested. And in the 5 eyes, it was just minute particles left at the equator of the lens, hidden behind the iris. So we have already some IP, some granted, some other filed, and we are in the process of filing some new IPs, additional new IPs. And we are developing a docking system to make the visualization more reliable and to have OCT probe in addition to the transpupillary OCT to see better at the equator of the lens. So this is pending. So despite the fact that we are spending all our effort and we are really focusing on cataract surgery, this technology can be potentially applied for other surgical fields. Regarding the competitive landscape, there are several robotic platforms available, but they are all except one used for other surgical application. Only one, the Precise, has been designed as well for eye surgery, but for retinal surgery, and they are not using any real-time guidance, and they are not dedicated for cataract surgery. So in that field, we are the only one. Regarding the risk, there are some technical risks. We think that being able to process the information in real time in all cases may be tricky, but the preliminary results are very promising. Some market risk with the barrier to adoption, but ophthalmologists in general are pretty good in the adoption of new technology. And the regulatory risk with maybe it will be difficult for the FDA to approve the system in a whole, but we think that with incremental FDA approval, it shouldn't be a problem. Regarding the timeline, so we need another 2 years to finalize the prototype. We have a proof of concept now, but we are building some additional features and making it more robust to be ready for animal trial in 2 or 3 years from now, and human trial hopefully in 3-4 years. FDA approval starting hopefully in 2024, and reaching market in like 4-5 years from now. Thank you very much. Okay, good.
Ali Reza. And I'll let Dr. Moshe Varinia introduce himself. I'll just get this up for you. Oh, yeah, sure.
Good morning, everyone. It's great to be here. I want to thank Dr. Savary for inviting me and including me in this program. I'm Ali Reza Moshavernia. I'm a board-certified prosthodontist, and I'm assistant professor, soon to be an associate professor, Division of Advanced Prosthodontics at the School of Dentistry. I'm going to talk about Dopamer, which is a novel bioactive dental filling material with remineralization power. So what is the problem that we are trying to solve, and what is the focus of this presentation here? We have more than 2.3 billion people globally that they are suffering from different type of form of dental caries. If it's not treated, it's going to cause pain, infection, tooth loss, and it's going to significantly reduce the quality of life of our patients. And if you look at the reason behind all these dental caries, not only the not having a good oral hygiene, but also most of the dental materials available in market for being used as restorative dental materials, they are offering some form of recurrent caries, recurrent decay, around 40% of them. So what we are trying to do with with my team here is we are trying to find a solution for this recurrent decay around dental filling materials by developing a new dental filling material which has remineralization power. In addition, it has some other advantageous properties that we're going to talk about. To start with, our team consists of myself. I'm a prosthodontist. At the same time, my background is in dental materials, stem cell biology, and tissue engineering. And Dr. Hassani, who has a PhD in chemical and bioengineering, and he's our like biomaterial scientist and tissue engineering that we work together. Before I start my presentation, I would like to share with you this quick video that talks about tooth death cycle. Most of you that you have feelings when you go to your dental office, the dentist is going to tell you that you need a filling. He's going to do a filling, and after you go there for a checkup, you might see that the dentist will tell you that you have a recurrent decay, the filling needs to be changed, and the next time you have to do a bigger filling for your tooth, and then sometimes your tooth is going to become sensitive because of the recurrent decay. Then root canal therapy needs to be done. They're going to put a post in your tooth, and at the end, the dentist will tell you, you know what, the tooth cannot be saved. We have to extract and place a titanium dental implant. So this is the death cycle for our teeth, and what we are trying to do, we are trying to avoid that and prevent that. If you look at the current dental filling materials that most of you are familiar with, they are either based on silver filling materials, we call it amalgam restorations. They are very cheap, they are long lasting, however, they have mercury cytotoxicity problem, the color is not good, and they do not have any remineralization power. And we have our resin products, resin composite resin materials that aesthetically they're fine, however, they don't have any remineralization power, and after a time we can have recurrent decay, and then if you see here, the fillings are going to change because of the decay, and they're going to fail, and you're going to have the problems, and we have to deal with that. In order to address this problem, we have developed a novel dental filling material. It's a restorative dental filling materials that it has remineralization power. So it's not only tooth colored, not only it can be used for like all the aesthetic restorative dental filling materials in everyday clinical practice, but also it prevents any decay, it prevents any remineralization, any hypocalcification or demineralization, and at the same time, it has antimicrobial properties. The technology is based on polydopamine, basically technology that we have utilized and we have added to the fluoroalbosilicate glass particles, and we have named the Dopamer technology because of just the dopa from the polydopamine technology that we use, and the ionomer presents off on the liquid part. So it's like the powder and liquid that can be mixed together, and it can be in different type of like dispenser. It can be injectable, it can be like pre-capsuled and mixed, and it can be used in different clinical applications. Right now, we are able to fabricate this technology. These are the Dopamer filling dental material at more lab scales, but we are going for more production and more going toward like semi-industrial scale. And what we have shown is in comparison to our competitive dental restorative materials available in market, the price and our expenses are significantly lower in comparison to commercially available products. Usually when you talk about dental materials, the first thing that dentists, they want to look at is like the mechanical properties. So we have utilized one of the gold standards in restorative dentistry that are being used constantly and routinely in dentistry, and we have compared some of the mechanical properties and handling properties like setting and working time in comparison to our dopamine technology. As you can see here, we have not only comparative but also significantly higher mechanical properties in comparison to our other commercially available products. Not only that, when we are trying to treat our enamel and dentin and we put our dentin or enamel in artificial saliva in vitro, we have clearly, and as you can appreciate here, show that we are remineralizing the surface of enamel and dentin. And think about it, for example, for this study that we have done here, if you have a dentin that it has already some form of caries, in presence of our dopamine technology restorative dental material, we are remineralizing the dentin basically structure. And as you can see here in our mechanical properties and nano-indentation studies, we show the significant increase in the nano-hardness of the material. So not only it basically prevents the dental caries, but also if we have any form of dental caries, it can cause remineralization in presence of artificial saliva and in our basically patient's oral environment. We have done a proof of a study in rats. So basically we have placed our dopamine restorative material in rats, and we have compared it with different type of restorative material, which shows like very promising results. And not only it prevents the dental caries, but it basically promotes dentinogenesis, basically fabrication and formation of new dentin structure in our teeth. What is the market potential? Actually, restorative dental materials in 2016, it was around 4.6 billion, and we're expecting to be around 7.2 billion in 2022. And if you look at the breakout, like a different type of materials available in market, how much each one is basically being used, you can see here like our composite resin and amalgam restorations. So we have a very good niche, and we have a market to work on that. We have already patented our technology. We have 2 more pending, actually patent applications follow up to this dopamine technology, and currently we are working with our collaborators for NIDCR and C-Doctor program for pre-FDA meetings to push this project forward. At the same time, this technology is not only being used as a restorative material, we have proof of concept, and we have studies that shows that this study can be used for anti-sensitivity products. It can be used for prevention of early dental caries, like white spot basically prevention. And also we are in the process of fabricating and making a new toothpaste that has remediation power for our patients. Just look at quickly at the competitor matrix. There are like many different type of dental material manufacturers in the market. However, if you look at this chart that we have here, our technology, dopamine technology, consists all the basic required properties that we need from an ideal dental restorative material that none of our competitors they have. They don't have mineralization power, they don't have antibacterial and anti-cavity properties, and they do not produce and exhibit the right mechanical properties that we want for everyday dental application. Yes, there are some risk and medication, and we have encountered some, and we solve them, and we are aware of like some other risks and we are working on them, not in terms of technical risk, but also market risk that we are going to address them. Right now, we are at the milestone 4. We have been able to optimize the mechanical properties, optimize the handling properties and the regenerative properties of this material. And with the help of UCLA Innovation Fund, we are trying to push this project forward and progress. And hopefully, by the end of this 2-year milestone, we are going to have a clinical trial, and after application for a 510 submission, because we have worked with our Division of Orthodontics here at UCLA School of Dentistry, and we are in the process of designing a clinical trial for our dopamine technology for our patients. And we are very hopeful, and we are looking forward to this collaboration and this clinical trial for our patients. And by that, I would like to thank you very much for your kind attention. If there are any questions, I would be happy to answer. Very different. Great.
Exciting stuff. So to wrap it up, I'm just going to put it's not working anymore.
I don't think so. Thanks very much. So everyone in the room, either themselves or has had a loved one who's come into contact with the orthopedic surgery world, whether it's a hip or knee replacement, or it's spinal hardware being put in, or it's fixation of a fracture, I think everyone can relate to that moment when you walk in and have an orthopedic surgeon like me say, sign this consent form for surgery. We're going back, and the number one risk you're signing about is that there could be an infection. That's the number one risk always. But do you guys and does the patient really know what that actually means? So let's talk about a hip or knee replacement that gets infected. So first it gets infected, they come back and we have to take out the implant. All of the implant and all of the surrounding bone have to come out. That's a major operation. There's then IV antibiotics for 8 weeks while the patient is more or less bedbound while their knee looks like this. There's no bend in the spacer that's put in there. There's then an antibiotic holiday of 2 to 3 weeks where the patient remains with that spacer. Then we get labs and aspirations and we hope that things are clear. And if they're a lucky one that's cleared, they get another major surgery to get a new implant put in. So that's down here on the bottom. So with all of that, this 2-stage process, about 3 months of time, roughly $160,000 of direct cost. The success rate of this 2-stage operation is 65%. So you could go through this entire thing over 3 months and then be back at stage 1 again. And then you're talking about repeating it or giving up your limb to eradicate the infection. So being diagnosed with an orthopedic implant infection has a 5-year mortality worse than being diagnosed with multiple myeloma, than HIV, than breast cancer. More than $8 billion are spent in the United States every single year on orthopedic implant infections alone. So what we've decided to work on is an effective, safe, and practical polymer that prevents this downstream catastrophe. And we believe the team represents all of the expertise needed to take it from the bench to the bedside. Our mission is to create surgical implants, to modernize surgical implants in the era of personalized medicine to prevent infection and patient suffering. The inventor team, myself and Tatiana Segura. Tatiana has since moved on to Duke University, but all of the IP is here at UCLA. So orthopedic infections, why is it such a problem? If infection gets on metal, it forms a biofilm. You have no vascularity. It doesn't matter how many IV antibiotics you give. It doesn't get to the implant because the implant doesn't have a blood supply. That biofilm over the top protects it from anything that you give locally. There's a 1 to 20% infection rate depending on the type of surgery, that 2 to 7% patient mortality, and again, $8 billion. So what do we currently do? Well, the last major advance in orthopedic implant infection was in 1972. In 1972, we figured out if we give antibiotics within 1 hour of surgery starting, we can decrease the infection rate. 45 years later, we're doing the exact same thing with not a single major advance. What do we do clinically with no data and no FDA approval? Well, right now we take vancomycin powder, like Parmesan cheese, that you'd put on pizza and you put it in the wound before you close and hope that works. No data to support it, not FDA approved, but hey, what else do we have? There's also bone cement that we use as a grout. We use cement in there to hold the implant in place. So we load the cement with antibiotics saying, hey, something's better than nothing. What's the problem with that? Well, cement is exothermic. It gets very, very hot as it hardens. That means it kills most of the antibiotic, so only a couple of antibiotics are heat stable enough to survive the exothermic process, and it traps 85% of the rest of the antibiotic in the polymer, so only 15% is getting out. The other problem is that putting antibiotics in the cement weakens the cement, so you've kind of lost the purpose of the cement in the first place. So what's the solution? Well, we've developed a point of application antimicrobial polymer. So it's applied in the operating room by the surgeon, and we'll talk about why that's so important. What does this do? It gives you a local high dose delivery of an antimicrobial. It has controlled release kinetics. We can release the antimicrobial over time as we wish to deliver it. It also has a smart trigger, and this is really important. Most developed technologies are passively eluting. That's a big danger for resistance formation. If you passively elute antibiotic, bugs eventually have time to work around it and develop resistance. If you give bursts of antibiotics at very high dose, you're above the MIC, which is what we often talk about, but you're also above the MPC, the mutant prevention concentration, that doesn't allow the bugs the opportunity to work around it. The point of application allows us flexibility in the operating room to decide what antibiotic we want to load. We'll talk about why that's important. This polymer is completely resorbed, and that is an important thing from regulatory side. There's cost conservation, which we'll talk about, and we believe a realistic regulatory pathway. Here's what it looks like. This is a human-sized nail being put on. You can apply it by spray or brush and then light cure it, and here's a coated implant. We took it to a well-known and utilized animal model that the FDA uses to assess efficacy, which is a mouse model of orthopedic implant infection. And you can see here in the green line that doesn't project out all that well, but absolutely no infection in the animal that has the polymer with antibiotic loading. What does that look like downstream here at day 42? You get this osteolysis in the uncoated implants or the coating without antibiotics, and you get no colony-forming units on the implant or in the surrounding tissue if you've used the polymer. So I won't belabor you with data, but 6 different animal models, 4 institutions across 3 countries, same results. Whether it's in spine surgery, whether it's in open fractures or hip and knee surgery, we've been able to prevent this downstream colonization, infection, and osteolysis. So if this is as easy as just sticking some antimicrobial on the implant, why are 45 years of no progress doesn't make sense. So what are the barriers? Well, there's cost barriers, right? The implants companies have a lot of pressure on them to keep costs of implant down. If you add a coating to it, it makes implant costs go up. Point of application avoids that. The surgeon now is getting this as a separate product and applying it, so you're not disrupting the chain of implant production. Implant shelf life. A lot of people have proposed putting coatings on at the factory. Then your implant becomes a biologic. It doesn't survive very long on the shelf, whereas orthopedic companies like to make these things, store them, and give them out when they're needed. Bacterial resistance we've talked about, and then the biggest is regulatory issues. We'll talk about how this avoids the classification of a drug delivery device, which has killed so many promising technologies. So what did we do first with point of application? Well, the first thing we did was we went to the people who would be our biggest consumers. We went to the Department of Defense, and we asked them for a grant. So they support us in the technology, in the development, because what they looked at is in Iraq, everyone was getting these blast wounds that were contaminated were all bacterial infections. In Afghanistan, the blast wounds were all fungal infections. It depends on the environment where a blast wound happens. So if you give them a coating with vancomycin on it or some traditional antibiotic, it works in some, it doesn't work in others. But if you now allow the surgeon at the time of surgery to add the antimicrobial that works for that local environment, you then give the versatility the Department of Defense needs to deal with the theaters as diverse as they are currently. Here, just a quick slide to show 10 common antibiotics all released above the MIC MPC with no modification of the polymer crosslinks. We've also used antimicrobials and antifungals and demonstrated and published this. So what else is out there? There's nothing approved in the United States. In Europe and in Japan, there are 2 technologies. We believe that when you start looking across at efficacy, ease of use, affordability, customizability, there's really nothing out there on the market that can compete with this. The market is really hard to estimate, honestly, because our belief is that if we have this, that patient demand will drive this. Once people know that there are antimicrobial coatings for implants, this will drive the industry forward. But these are our best estimates, and as we talked about, that $8 billion implant infection rate, that's what we're looking to prevent. So hospitals that are now being told that an implant infection is a never event and they're going to absorb some of that cost on themselves. If you get an implant infection at UCLA Hospital is supposed to absorb some of the cost of treating it. Well, then it becomes a much more desirable product to have to prevent that downstream catastrophe. We do believe it'll be reimbursable, lower readmissions, improve patient outcomes, and improve physician performance. So our timeline is here. We're in the GLP synthesis timeline. We are doing large animal safety testing now, and I'm happy to answer any questions. Thank you very much. Yes? Yeah, we have. So we have shown completely the efficacy is identical whether it is stainless steel, titanium, or cobalt chromium, which are the three main metals used in orthopedic implants. We have not yet done polyethylene, which is the most common plastic used. Our Tatiana, who's the chemist on this, believes that it will be equivalent, but we haven't shown it, mostly because the animal model we use is our sort of bread and butter FDA animal model, is a mouse model, and getting the implants out of the polyethylene has been more challenging. They're just so small that we haven't customized the implants, but we've done the three major metals, plastic to be determined. Any other questions? Yeah, thanks. So yeah, I sort of intentionally avoid it because it scares people going into the operating room, and I'm an orthopedic surgeon. But no, the incidence of infection, so in routine hip and knee replacement, it's just under 1%. So it's somewhere between 0.5% to 1%. So the odds are still very good. Now, with that said, I'm an orthopedic oncologist, so I take care of kids who are on chemotherapy, and we're putting in big implants, not just hip or knee resurfacings, but replacing whole bones. So in our patient population, the international data says 20%. The national data is around 10%. So these are real problems, and that's what got me interested in this field to start. Also, the other one with kind of that 10% is in the blast wound population. A persistent infection that's not cleared with multiple surgeries is over 10% for the military. Thanks.